Let me share an interesting fact today. According to research, that diatoms are also responsible for triggering the ice formation in the clouds. So you can see wherever the plant is being there, it is responsible to facilitate life through rains or something. Exceptional, isn't it? Hello Shishas, welcome to lecture 4 on cell zone 3. As the cell has revealed itself as a living unit, the mystery lies even today how the various chemicals in cytoplasm are able to produce a life, and it's been a long study for the scientific community to identify and replicate in the artificial conditions. The cytoplasm properties are it's a colorless fluid and homogeneous nature, amorphous and colloidal. So this allows the substance or the cell organelles to survive in the safe condition. And we have two various regions of the cytoplasm. One is called as the ectoplasm and the one is called as the endoplasm. And you can see the cell organelles alone will not form the cytoplasm. That's why the cytoplasm without cell organelles is called as the cytosol. The cytosol is being studied to understand how the cell organelles can survive. The cell organelles are nothing but the smallest structures found in the cell. They may have the membranes or they may not, but still they do the specific functions. So all your learning goals in this chapter is you have to know the specific functions, the structures and its names. So you're not doing more than that. Okay, so now we have classified the cell organelles, the microscopic functional units, based on the membranes. We have various types here. The few organelles are there which they have the double membranes or double membranous, or they have two membranes. The first one is the nucleus, mitochondria, and then the plastids. So these are the example. And we have single membranous organelles. The single membranous organelles include the G oral system, Golgi apparatus, Endoplasmic Reticulum, and Lysosomes. And then you can also see some other structures like the microbodies, which are smallest particles, are also the single membranous structures. And then you can see the non-membranous structures, the organelles that completely lack the membrane or the outer covering. And finally we can see the cell inclusions, and then we'll continue with the disorders of cell organelles in humans particularly. Alright, so let us begin with the double membranous structures. In the previous lecture we learnt about the double membranous structure, nucleus. We know that nucleus is a double membranous structure and it has various parts in it. The 4 different parts are already studied, and that's why I'm skipping that part now. We'll be learning about the mitochondria and then the plastids. The mitochondria are called as the functional units of the unit cell, where they particularly involve in producing of energy. And the plastids. So plastids are nothing but the structures found only in plant cells and certain blue-green algae and algae species to produce the energy by themselves through a process called photosynthesis. And even we can call these two as the semi-autonomous organelles. The label semi-autonomous organelles given for these two organelles, the reason is they have their own DNA. That doesn't mean they can be able to go like as a wish. They always under the control of nucleus, but still due to presence of their own DNA, so they can be able to replicate their numbers anonymously without the permission of nucleus, whereas this is a rare condition inside the cells. So that's why it is called as semi plus autonomous, the partial control over themselves. And this semi-autonomous nature is really a tough part to explain for many scientists in those days. But still, we believe there is a theory to explain how these organelles alone got their own DNA to replicate. So it's nothing but a cell inside a cell. You can compare like that. The cell which has the own DNA, again the interior organelle, they have their own DNA. That's why it is called as cell within a cell. And then in order to explain this semi-autonomous nature, a theory arose and which was most successful. So that theory is called as Endosymbiotic Theory. And the Endosymbiotic Theory was given by Dr. Margalis. So she actually proposed this Endosymbiotic Theory, and she tried to define the Endosymbiotic Theory like this. It is believed that before the evolution, some cells got engulfed into the organisms. When they engulfed by these organisms, they are able to engulf certain blue-green algae. So let's say there is a protozoan or the primary cell, which has the structures, and there is blue-green algae. So the nature of blue-green algae is it has the plastids. That's why it is blue-greenish in color. It has plastids, and then it has its own mitochondria. Whereas the mitochondria is completely absent in the primitive bacterial types. They have only mesosomes. And then it is believed that the ancient bacteria, this thing got taken blue-green algae or engulfed the blue-green algae. Then you can see the plastids which entered inside, and then mitochondria which entered inside. They became symbiotic for the organism. Okay, and then again we can see a condition. So when you say they are symbiotic in nature, again this cell got evolved for few million years. So few cells only survived with the plastids and mitochondria, and few cells only survived with the mitochondria alone. They lacked the protoplastid due to some genetical changes. Now we can call these structures with specific names. Okay, the organisms that have the plastids and mitochondria survived are called as the plant cells. The cells that only survived with mitochondria are called as animal cells. So these she proposed this theory, and it was widely accepted, and it gave a big name for discovery and explanation. Let us go back to the portion. So we're going to learn about the mitochondria. Remember, the semi-autonomous organelles as endosymbiotic theory is quite important for your NSCJS perspective. Next we'll look at the mitochondria, the first double membranous structure. The mitochondria is also called as the powerhouse of cell. It is found in eukaryotes, not found in the prokaryotes, and here you can see there is a specific cycle of events that takes place or called as the Krebs cycle. The Krebs cycle is generally found, now we can come back to the illustration here. So if you look at the illustration, you can find out it almost appears like a small unit. You can see two layers, inside and outside, and then you can also observe the layers, the folds inside. So this is called as the cristae, the inner foldings. And then you can see the ribosomes here, its own ribosomes. So exceptionally this is 70s. So when they have 70s, it is a big clue for the scientists that it is purely a prokaryotic cell, alright, or prokaryotic cell component. And then you can see the oxysomes here, the center of ATP synthesis, and the inner chambers. So the inner chambers will have the liquid region. So it is filled with the liquid, some proteins and enzymes that help in production of ATPs. And then you can see the inner membrane and then outer membrane. Okay, so these are the mitochondria looks like inside the cell, and it plays a major role in producing energy. It involves in a process called the respiration. The respiration is a two-step process in most of the organisms. So what happens is, the first step, the organisms are going to break down the glucose. So the breaking down of glucose is called as glycolysis, happens in cytoplasm of the cells. And then we have the other region. We have whenever it is an aerobic respiration, that means if it is oxygenated respiration, then only the cytoplasm is going to break down the glycolysis, and then the glycolysis will give a product called the pyruvate. the pyruvate will be going to come to the mitochondria for producing ATP. And if it is anaerobic, so let's say the respiration is happening due to carbon dioxide, so here you do not see the production of ATP molecules. Instead it produces the alcohol. So one of the commercial process, it helps in making the beverages. Sorry. So that's about the mitochondria, and then you can see small structures inside here, so which are called as F0 F1 particles. They almost appear like the serial light bulbs. So this region is called as F0 and it is called as F1. So here you can see the Krebs cycle is occurring. So don't get confused. Krebs cycle is involved in producing of ATP molecules. Even it has other name called the citric acid cycle. Clear? Let's go back to the second structure. So second structure is okay, I think I missed with this one. The mitochondria are in more proportions in cell organelles, like in some organs like the liver, where it will be around thousands of numbers. I only mentioned 500 here. Depending on the action, depending on the purpose, they will be producing a lot of energy. And in also kidneys you can find more number of them. Okay. But you can see in case of yeast, one of the fungal type, it has less than 10, so that means it won't produce more energy. Okay, and then if you look at the functions, it helps in the aerobic respiration, yolk formation, elongation of fatty acids of the cell. Okay, let us look at a second important part, that is the plastids. You might have heard about the chloroplast for a long time. So chloroplast is nothing but a type of plastid. Based on the color they have, we have classified the plastids into two major groups. First group is called as the leucoplasts, that are colorless, and then the chromoplast, that is colored structures. The leucoplast main function is to store various substances. You can see based on what they store, we have named them accordingly. First one is called as the amyloplast, that is starch, and then we have the second type called proteinoplast, that stores the proteins, and then we have elioplast, that stores the fats. So these are mainly involved in storing of food, and even they can be able to undergo the color changes. And second thing is the chromoplast. The chromoplasts are interesting structures. Whatever the colored regions of the plants, they are nothing but the chromoplast. Based on what color they produce, we have classified them into various names. So first you can see the chloroplast, that's nothing but the green color, and then the rhodoplast, it's nothing but the red color, and then the brown is nothing but pheoplast, and keratin also a type of pigments. And next is, keratin is also like they give orange or brown color pigments. And next is the chloroplast and its importance. Let us try to look at the structure of the plastid. And again, I told you, as per the endosymbiotic theory, it has very close association of own ribosomes like 70s, and it has a matrix, and then you can see the inner outer membranes, and these are very important structures called granum. So they have the stacks of thylakoids. So when you say stacks of thylakoids, you can imagine the piles of the coins. It almost appears like this, and one unit of this is called as the granum. Okay, all the units are called as grana, and then you can see it appears, it's like flattened, that's why you can see kind of lines, and then you can see the granum and then stroma are very important regions. So the stroma is a place, I think the matrix I mentioned, so stroma is a place where an important set of reactions of photosynthesis takes place. It is called as the dark reaction. And then the granum is a place where the light reaction takes place. So light reaction is nothing but where the ATP and NADPH2 are produced, and then the dark reaction is responsible for conducting or occurring a step called the Krebs cycle. Oh, I'm sorry, it should be the Kelvin cycle. So these are very important. So dark reaction without light and light reaction with light, ATP and NADPH2 are produced, and here the Kelvin cycle uh, occurs here. So this is about the structure of the chloroplast, and now we'll go back to the functions. The functions of photosynthesis, they help in photosynthesis and they help in storage of materials, particularly leucoplast and then chromoplast. And sometimes you may get few questions quite relevant to this. When there are few vegetables, they undergo some decoloration. So this decoloration due to when the chromoplast is gradually turning out into various pigmentations. Particularly in the beginning stage, you can see the tomato always appears in the green color, but as it matures, due to some external effects or external conditions, it undergoes some color change. So it's due to chromoplast or undergoing few changes inside. Alright, so that's about the first set of organelles, that is called as double membranous structures. And now we look at the single membranous structures. The single membranous structures are as follows: the GE-oral. So remember, the GE-oral is nothing but a endomembrane system. It is a specific system that includes three different cell organelles, that is Golgi apparatus, endoplasmic reticulum, and then the lysosomes. The lysosomes are also called as the suicidal bags. The main function of lysosome is they have a lot of digestive enzymes, so they help in the breakdown of organic matter. And also the lysosomes also help in a process called apoptosis. The apoptosis is a process where when cell is attaining certain age, so the internal environment of an organ, they always prefer to make the cells renewed each and every day. You do not know how many cells inside your body are being renewed. So whenever the renewal process occurs, the cell which got older enough, so they undergo a process called apoptosis. It's nothing but the suicide of the cell. The cell is going to die itself, and lysosomes play a very important role in that. And next is the Golgi complex. So the Golgi complex is a other structure which produces the Golgi bodies, and their smooth, flattened, sac-like structures called cisternae, and even they have certain vesicles. Vesicles you can compare to the bowls or dishes you can see in kitchen. So they help in moving of substances inside the cytoplasm. And we have two types of vesicles based on when they bring in or they take out. So it's cis and trans, and also the main function of this is to produce or repair the membranes, and also they involve in secretion of certain passage substances. Now we look at the illustrations after endoplasmic reticulum. The endoplasmic reticulum is one of the important structure for the cell, because it is surrounded by it surrounds the nucleus based on the regions. Let's say the inner and outer cell membranes. It has two types, the rough endoplasmic reticulum and smooth endoplasmic reticulum. The rough endoplasmic reticulum has ribosomes, hence it is called as, the surface appears like rough. And then smooth endoplasmic reticulum doesn't have any ribosomes, hence it is smooth in nature. And mostly it helps in producing of the fats, and then it helps in the steroid production that gives the male and female characteristics. So the important function of endoplasmic reticulum, remember this, it helps in the production or biogenesis of cell membrane, and also they detoxify most of the toxins, and they play major role in various metabolic reactions. Alright, so let us look at some illustrations to understand this. Here you can try to test yourself whatever you learned here. You can observe the Golgi apparatus here, the endomembrane system, the Golgi apparatus. So these are called as cisternae, smooth, flattened, disc-like structures you can see, and it has cis, that is going towards the nucleus, and you have trans, that is going towards the cell membrane. And mostly, if you can recall in previous things I told you about exocytosis. So mostly this is an example of exocytosis here. So how the proteins are getting expelled out of the membrane. And then you can see here the endoplasmic reticulums as well, the rough endoplasmic reticulum and then smooth endoplasmic reticulum. So here the smooth ones, and then there the rough endoplasmic reticulums, very important for cell functions. And these only endoplasmic reticulum, so the nuclear pores, and they are very close to the nucleus. Now we'll go back to the next regions. So next single membrane structures are nothing but the microbodies. The reason why we call microbodies, maybe it's due to the smallest structures inside the cell, and they also play a major role. So first one is called as the peroxisomes. The peroxisomes in animals, they help in peroxide metabolism. So peroxide is one of the effective oxide that can be able to oxidize a lot of substances. That's why peroxisomes will specifically involve in the H2O2 metabolism. And also for plants, it helps in a process called photorespiration. The photorespiration is not an useful process for the plant. We knew that plants actually take CO2 inside, they make convert it into the glucose. So when this process is happening, the plants have a problem. You know that plants also produce the oxygen. So whenever the oxygen is being deposited inside the plant, so it doesn't allow the plant to undergo for the photosynthesis. Hence, the photorespiration happens in order to get rid of oxygen being evolved inside the plant. That's why this process is called as photorespiration. In the presence of light, respiration is taking place. And next is called as Sparrows. So Sparrows is also a site in the plant cells. Majorly, it helps in the lipid synthesis and storage. And then we have glycosomes as well, so which are almost similar structures. They're mostly found in the oil containing seeds like castor. Sparrows will be helpful in two purposes. They produce lipids as well as storage, but mostly the glycosome structures might be responsible for producing such fats. So the next structures are nothing but microtubules. The microtubules are the eukaryotic cells. They have the special protein called tubulin, and these are found in cilia, the hair-like structures, and flagella, the locomotory structures. You can see nucleina, and then the centrioles, and in cytoplasm as well. So centrioles we're going to learn soon, and the cytoplasm require the skeleton. So the skeleton will be mostly provided by the microtubules. So without microtubules, the cytoplasm may not be a specific structured organelle. So it could be, if it doesn't have any skeleton, you knew what happened. So it will not give a proper support. So finally, we're going to look at the vacuole. So vacuole is a structure that is a fluid filled structure. You can see it is covered with a membrane called tonoplast. So tonoplast is the only membrane outside, and this also plays major role in plants. Mostly it may be absent or present in a smaller size in case of the animals. So tonoplast is a structure that covers the membrane that covers the vacuole. And now we have reached almost the last topic, that is the non-membranous structures. So before going into that, let me show you the structures please here, and then you can also see how it's arranged inside the flagellae. Alright, so now we'll continue with the last structure. The non-membranous structure is ribosome. So ribosome is most smallest structure, one of the smallest structure in the plant cell, and it has very rich RNA and proteins, and it always made of two unequal subunits. So this is an important region you have to focus, because you can easily get confused. Whenever you say the prokaryotic cell, it has 70S ribosome that is made up of two unequal subunits, and when you say the eukaryotic cell, it will have two unequal subunits called 60S and 40S. So you should never supposed to we're looking at the the ribosomes. The ribosomes are quite different organelles inside the cell. You can see it has the two different subunits. I'm trying to illustrate you how the protein synthesis will take place, which is not part of your uh, syllabus, but still you can understand the ribosomal structure here. So when you see here the ribosomal structure, you can see two different units. One is large subunit, and another one is a small subunit. The large subunit is responsible to accommodate the various the transfer RNA subunits, and you can see the smallest subunit will be responsible to produce the proteins. That's how it helps in making of the proteins. Alright, let us go back to the next part, and you can see the centrosome. I think already elaborated a bit here. So the centrosomes as the structures are two microcylinders that are present in the right angles. So they always lie in the right angle. That's why when you try to illustrate the cell division, they always been represented in this way. Okay, and then finally the major function of the centrosome is to form the spindle fibers, and also they are the basal bodies of cilia, flagella that form the cytoskeleton. And finally the cell inclusions. The cell is not an idle thing, so it always works for longer time. Whenever it works, it always leaves some of the temporary non-living substances. It may be excretory, or sometimes it's minerals. So these structures are called as the paraplasm or deuteroplasm. And then the flagella and cilia structures that help in locomotion. So here, that is the end of the cell organelles and its functions. And now you can see the whole structure of cell. So you can see the cytoskeleton, the deeper layer, and then the secretory vesicles. The cytoskeleton again have various portions. Okay, I think it's an image, it won't accept it. And you can also see the microtubule here, and then the intermediate filament, the microvilli, and that help in absorption, and then the centrosome, the various types like centrioles or the right angle things. Okay, and then the plasma membrane here, the lysosomes, like deeper, then smooth endoplasmic reticulum, rough endoplasmic reticulum, Golgi apparatus, and then actin filaments. And you can see in the cytoskeleton how they are arranged as arranged inside. And then you can see the cytoplasm, then mitochondria. So the central nucleus, the nucleolus, nucleoplasm, and then nuclear pores, the double membranes you can clearly see here. So it's about the cell. And when you come to the disorders of cell organelles, cell organelles are one of the microscopic units. When they get damaged due to some reasons, it always leave a lot of disorders. For example, the mitochondrial disorder, that is eyes. So whenever the mitochondria are not properly being maintained, the mitochondrial number are not being maintained by the nucleus, so it leads to the visual loss and blindness, and even in case of brains it may cause the seizures. And then the lysosomal storage disorder. So it is one of the well-known disorder. You might see in newspapers a lot of people asking for help to get treated. So it happens one in a million, but still the reason for this disorder is due to accumulation of nutrients inside the lysosome. We studied that the lysosomes have a lot of digestive enzymes. So when these digestive enzymes are not actively breaking down the nutrients, it obviously going to leave a lot of nutrient storage. That's why it is called as lysosomal storage disorders, and this gradually cause death of the person as well. Alright, so I'm going to tell you a brief introduction about the cell division, and after that we can continue with next lecture. So here the cell division is a process where the cell wants to maintain its size. Here you can see the cell division is nothing but reproduction of its surface, and there are three different types of cell divisions. So first one is called as amitosis, given by Remark. So here the cell division takes place without a specific change in the nucleus, so it's like kind of fission. And here examples are blue-green algae and yeast budding is replicating division, so it produces exact copies of the things. So here you can see first time it was discovered in case of animals by Fleming, and then in case of plants by Strasburger, and it is divided into two phases, the cytokinesis and karyokinesis, as I told. But based on the life cycle of cell, we have classified them into two major steps or stages. So first stage is where the cell is going to uh, maintain its growth. So once it finishes with maintaining its growth, and it goes for the division. So that's what you can see here, the first substages of interphase. Interphase is also called as preparatory phase or cell cycle. That is a period between two successive divisions. Okay, so we have three different steps here: the G1 phase, S phase, and G2 phase. The G1 phase is one of the longest phase, and it is metabolically active, and you can see this is also called as pre-DNA synthesis phase. Now why we call pre-DNA synthesis phase, because in the S phase, the G refers to the gap. Okay, these prefix G refers to the gap, S refers to the synthesis. So when you say synthesis, it helps in producing the duplication of DNA will takes place here, and then it will have the centrosome replicated already, and finally we have the G2 phase that helps in producing of tubulin, and the spindle formation will be also on the way. So when cell is undergoing for cytokinetic division, and energy will be stored in G2 phase for the ATP production as well. So the cell functions may be temporarily ceased due to nucleus is under division. And second phase is called as the division phase. So the division phase is where the it is divided into the karyokinesis, division of nucleus, and cytokinesis. If you compare the mitosis with amitosis, the major difference between those two is it completely lacks the it completely lacks how the cell division uh, cell division is going to happen. You don't see any kind of changes. As soon as it gets some nutrients, it's going to duplicate. But whereas in case of the plants or animals, it waits for few steps. So interphase, the three steps: non-dividing growing phase. So it has G1 phase, S phase, and G2 phase. Then you can see the M phase that has two: short and dividing phase, karyokinesis, and then the cytokinesis. The next lecture we're going to learn about, so karyokinesis, it has four different stages and how these four different stages are seen. And then cytokinesis is only one step, but it is different in case of plants and then animals. So that's what we focus in the next lecture.